Welcome back to the Abattoir. I am Brian Cross, Associate Professor of Biology at Tulsa Community College, and your host for this very special COVID-19 Super Pandemic edition of Wide World of Dissection. Last time we began our study of the phylum Mollusca by examining the squid. This time we continue our exploration of this phylum by examining another fascinating creature, the freshwater mussel. Although a member of the same phylum, this bivalve, or two-shelled mollusk, presents a stark contrast to the cephalopod. The squid was a complex and sophisticated predator, living and dying by its wits, doing battle with marine creatures both great and small, going at its enemies hammer and tong, beak and radula. On the other hand, the bivalve mollusk is a peaceful filter feeder, and a contender, along with the sponge, the jellyfish, and the koala bear, for the title of stupidest of all animals. That doesn't mean that the clam, the oyster, the mussel are not successful. They most assuredly are. But they do ably illustrate the lack of correlation between cognitive ability and reproductive success, a phenomenon that has bedeviled the scholarly across the world for untold ages. Lucky for us, the lack of a brain does not make them less intriguing as a subject of study. So prepare yourself for a trip to the zoological antipodes. I present to you the freshwater mussel.
Now here we have an example of a bivalve, or two-shelled mollusk. As previously noted, the bivalve mollusk does not have a head, does not have a brain, does not have complex behavior, as befits an organism that makes its living by filter feeding. But as you can see here, there is one way in which the bivalve mollusk shows sophistication and refinement, and that is in the enjoyment of a fine Cuban cigar. This individual was so devoted to it that it refused to give up its cigar even upon death. Now, our first look at a bivalve mollusk can be rather confusing, just as it was with the squid, because just by looking at it, it's very hard to tell what's the top, what's the bottom, what's the anterior end, what's the posterior end, where's the mouth, where's the butt, what's left, what's right. And so just as we did with the squid, we're going to, with this bivalve mollusk, begin by orienting ourselves to the cardinal points of direction. So we have over here on the left side of your screen, we have the anterior end. And over here on the right side, we have the posterior end. Now up here, where you see this little protuberance, the umbo, you have the dorsal side. And down here where the shell is open, the outer edge there, you have the ventral side. Now the way I was able to tell the difference between the anterior and the posterior ends was that umbo again. So the umbo is up here on the dorsal side, but it is offset towards the anterior end. So you see it's kind of turned towards the anterior end, and so that tells me which end will have the mouth, which end will have the labial palps, as we'll see in just a moment. Now if you put that all together, you'll realize that these shells are not top-bottom shells, they are rather left-right shells. So this one over here is the left shell, and this one over here is the right shell. The midline goes right in between the two shells. So that is the mid-sagittal section there between the two shells. So if we flip the bivalve over so that the ventral side is up, we can see that the shells don't just fall open. Right? They're held together very tightly by the adductor muscles. To adduct means to pull in towards the midline. So the adductor muscles for this bivalve mollusk pull in the left and right shells towards the midline, keeping the shell closed. Now there are two big bands of muscle. There's one at the anterior end, right here, and there's another at the posterior end. So we will need to cut through those big bands of muscle if we want to open up the shells and see the mantle cavity and the central visceral mass. Now before we cut the adductor muscles and open up the mantle cavity, let's look once again at the umbo. The umbo is this little protuberance on the dorsal side of the shell. It helps us to distinguish the dorsal side from the ventral side, and also the anterior side from the posterior side, because remember the umbo is offset a little bit towards the anterior end. Now the umbo is the place where the shell first begins to grow. So it starts there, and then the shell is extended outward in successive bouts of growth underneath the umbo and extending out beyond the umbo. So as we look across the shell here, we see lots of little ridges. Each of those ridges represents a bout of growth during which the shell was extended. Okay, so now we're going to open up the two shells of this bivalve mollusk and get a look at the mantle cavity. I've already cut those adductor muscles that hold the shells together, but we need to sort of very carefully peel back the mantle. So I'm going to speed this up a bit so we can get to the good stuff more quickly. You can see the siphons back here. Okay, right there, and you can see the foot peeking out. You can see a good view of the mantle there lining the shell. We're going to just very gently peel this back now so that we can take off the left shell. Right, and we'll pause it here for a second so that we can see some of these structures. You see the adductor muscles there, a big band of muscle there. You can also see a good view of the mantle, very thin towards the middle, very thick out towards the edge. You're seeing the left side of the mantle there. I've removed the shell, but the left side of the mantle is still present. Okay, so now let's get farther into the mantle cavity here and take a look at some of the other structures. So we've peeled back the left side of the mantle, so there's the right side of the mantle, and the left side has been peeled back. It's at the top of your screen. So let's look at some of the structures. This is the mantle cavity, the space enclosed by those two layers of the mantle. Back here we have siphons that will bring water in and shoot water back out. That flow of water is necessary for feeding. We have the gills here that will catch the food in a layer of mucus, labial palps that will pick up that food and deliver it to the mouth. The gills, of course, are also involved in oxygen. Down here we have some dense tissue. This is the dense spatulate foot that is used for digging in the sediment. It's the densest tissue here, except for those adductor muscles. If you go up dorsally here, right, dorsal to the spatulate foot is the central visceral mass, which is a bit spongier. We'll cut into that, split it in half here in a moment. Okay, so just to review some of the structures of the mantle cavity here, we have the foot, the spatulate foot. That's this dense mass of tissue here that's used for digging in the ground. And then we have the labial palps here, and those labial palps are used for picking up the food from the gills and putting it into the mouth. And then we have the gills themselves here, these thin flaps of tissue. Now remember, all these structures except for that spatulate foot are going to be bilaterally symmetric. So you'll have two labial palps on the other side, you'll have two flaps of the gills on the other side. Now note that the gills are here right in front of the excurrent and incurrent siphon. The excurrent siphon is dorsal, the incurrent siphon is ventral, and so the water is going to come in, flow through the incurrent siphon over the gills, through the mantle cavity, and then back out over through the excurrent siphon. And that flow of water is going to bring oxygen that the gills will pick up. It's also going to bring little pieces of food that will get stuck in the mucus on the gills and then directed forward. The mucus will flow forward towards the edge, ventral edge of the gills, and forward where it will be picked up by the labial palps. You also see a good view of the mantle here. You can see the mantle of the right side peeking out, the thick edge of it, and then you have the mantle from the left side here folded up. Mantle is going to secrete the shell, and it will function in gas exchange, and it creates these siphons and this mantle cavity within which so much happens in the bivalve mollusk. Okay, now before we look at the internal anatomy, we're going to turn the mussel over here and look at the dorsal surface. And you can see here on the dorsal surface, after we get a few pieces of shell out of the way, that there is this thin-walled cavity here, and that is the pericardial cavity. Now, as you might already know, the pericardial cavity surrounds the heart. That's literally what pericardial means, is surrounding the heart. So if we peel back that thin-walled cavity, the membrane, we can see the heart of the mussel. Now here's a view of the same structure. This is the same dorsal aspect of the mussel here. You can still see the heart here, but you can also see more of the surrounding structures. In particular, you can see this very, very strange pattern that we have in bivalve mollusks. Truly, it can be said that the way to a bivalve's heart is through its stomach, because the intestines actually pass right through the middle of the heart. So here's the heart, and here's the intestine before the heart and after the heart, anterior to the heart and posterior to the heart. So the intestine winds around down there through the central visceral mass and then proceeds dorsally, passes through the heart, and then after passing through the heart, it discharges the fecal material into the excurrent siphon. Okay, so the last thing we need to do here is to look inside the visceral mass. So I'm going to cut off a bit of the foot, and then we'll do a sagittal section through the rest of the foot and the visceral mass so that we can splay this out and get a good look at the internal anatomy. So here we go, pinning it down, and there you go. So this is the midline right here. So you've got the right side to the top and the left side to the bottom. Right here is the mouth. Okay, on both sides, then passing down the esophagus into the stomach, and then the intestines wind around down through the digestive gland. So the green material is digestive gland, and then of course it's going to pass back through the heart dorsally, and then out the posterior end, discharging through the excurrent siphon. Okay. Now, before we wrap up, we need to say a word about the reproductive biology and typical life cycle of the bivalve mollusk. Most bivalve mollusks are gonochoric, that is, dioecious, with separate males and females. So males producing sperm, females producing eggs. Both are released into the external environment, and fertilization will happen out there away from the parents. That will form a zygote, which develops into an embryo, which then develops through stages like the trochophore larvae and veliger larvae, which we've already encountered. This is the typical pattern for the bivalve mollusk. But in some bivalve mollusks, we have a much stranger life cycle. In particular, in some freshwater mussels like Lampsilus, we have a life cycle that incorporates a significant amount of parasitism. In these species, fertilization is internal, so sperm is drawn into the female's mantle cavity, fertilization happens there, and then the female broods the offspring for a while. The offspring will then develop into this larval stage, the glochidium, and the glochidium is adapted to attach to the gills of fish. Here you see some little glochidia. These glochidia are adapted to clamp down on fish gills. Here you can see an image of fish gills with these little tiny circles attached to them. Those are the glochidia. They're going to feed off of the fish gills for a period of time, and then when they're ready, they'll drop away, they'll release, drop away, they'll set up shop on a substrate, attach to a substrate, develop into adults, and then filter feed for the rest of their lives. The strangest part of the story is how the glochidia get to the fish gills, because the mothers are not going to leave this up to chance. They're going to produce a variety of different kinds of lures that will bring in the fish species. Here you can see a very convincing lure from a species of Lampsilus. This will attract in the appropriate host fish species. When that fish approaches, the female will then spew out all of the glochidia. The glochidia will attach to the gills of the fish and then be carried away. This can be an important adaptation in some of these freshwater species. In particular, it allows them to move against the flow of the stream, so they can hitch a ride on those fish, get resources from the fish, but also they can move upstream rather than just getting pushed constantly downstream. Quite an interesting adaptation here with some of these freshwater mussel species.
Well, that's it for our tour of the mussel. I hope you've enjoyed your encounter with this dim-witted denizen of the deep, the bivalve mollusk. Next time, we'll begin our exploration of the phylum Arthropoda, the most speciose of all animal groups. Thank you for watching. Stay well.